To master the ecology sections of competitive exams, we must move past broad concepts and focus on exact data. Our objective here is extracting, categorizing, and mapping the precise numerical and geographic data that defines global biodiversity. We start with the term itself. The concept of biodiversity, popularized by sociobiologist Edward Wilson, describes the immense heterogeneity that exists across all levels of biological organization. This heterogeneity is structured into three specific hierarchical levels. You must categorize data into genetic diversity at the base, species diversity in the middle, and ecological diversity at the ecosystem level. Mastering this categorization is a prerequisite for the assertion reasoning portion of the exam. You will eventually use these three distinct levels as the baseline variables to evaluate how biological variety influences the overall stability of an ecosystem. Looking at the first tier, genetic diversity, we measure variation within a single species. In India alone, we catalog over 50,000 genetically different strains of rice and 1,000 varieties of mango. We also see this in the medicinal plant Rauwolfia vomitoria growing in the Himalayas, showing high genetic variation in the concentration of the active chemical reserpine. Moving up to species diversity, the metric shifts to variety at the species level within a specific region. The standard comparative dataset requires knowing that the Western Ghats possess a statistically greater amphibian species diversity than the Eastern Ghats. Finally, ecological diversity measures variation at the biome level. India features extensive ecological diversity, encompassing deserts, rainforests, mangroves, coral reefs, wetlands, estuaries, and alpine meadows. Compare this vast biome variation against a Scandinavian country like Norway, which has significantly less ecological diversity. Genetics, species counts, and distinct biomes operate simultaneously. Together, these three independent variables accumulate over millions of years of evolution to create the biological heterogeneity of the planet. Currently, the total recorded global species count sits at slightly more than 1.5 million. Within this global inventory, India accounts for about 8.1% of the total species, strictly defining its status as one of the 12 megadiversity countries in the world. Animals make up more than 70% of global species. Zooming in, insects comprise 70% of all animals, 7 out of every 10. Notably, fungal species outnumber all vertebrate species combined, exceeding fishes, amphibians, reptiles, birds, and mammals. The sheer volume of terrestrial invertebrates and fungi frequently contradicts common assumptions, which tend to favor large mammals and birds. Because of this blind spot, these exact percentages appear frequently as trap questions on standardized tests. Biodiversity is not uniformly distributed. It follows a strict latitudinal gradient. Global species diversity systematically decreases as you move away from the equator. We track this using specific avian datasets. Colombia, located near the equator, harbors nearly 1,400 species of birds. India, largely in tropical latitudes, holds more than 1,200. Move to temperate New York at 41 degrees north, and the number drops to 105. Reach Greenland at 71 degrees north, and you find only 56 species. Ecologists propose three primary hypotheses for this high tropical diversity. First, temperate regions suffered glaciations, while tropical latitudes remained undisturbed, providing time for speciation. Second, tropical environments are less seasonal and more constant, promoting niche specialization. Third, the tropics receive higher incident solar energy, driving higher productivity. All of these factors culminate in the largely tropical Amazonian rainforest in South America. Here is the ultimate data set you need: 40,000 species of plants, 3,000 fishes, 1,300 birds, 427 mammals, 427 amphibians, 378 reptiles, and more than 125,000 invertebrates. Geographical stability and continuous solar energy act as a massive incubator. This specific combination renders the Amazon the baseline for maximum planetary biodiversity. As explorers survey larger geographic zones, they discover more species. But this relationship is bounded by strict mathematical limits. Species richness increases with explored area, but only up to a point. This Cartesian graph plots the species-area relationship as a rectangular hyperbola. The z-value slope is typically 0.1 to 0.2, but for entire continents, the slope steepens significantly. For tropical frugivorous birds and mammals, it locks in exactly at 1.15. These z-values serve as strict mathematical parameters. They allow ecologists to accurately predict how rapidly biodiversity scales over massive geographic expanses. The current rate of species loss is severely accelerating. According to the IUCN Red List from 2004, we have documented the extinction of exactly 784 species, comprising 338 vertebrates, 359 invertebrates, and 87 plants over the last 500 years. The required examples of recent global extinctions include the dodo of Mauritius, the quagga of Africa, Steller's sea cow from Russia, and 3 specific tiger subspecies, the Bali, Javan, and Caspian tigers. You must also recognize the thylacine of Australia, seen here, as a prime testable example of recent vertebrate extinction. There are 4 major categorical causes of this accelerated biodiversity loss, collectively termed the evil quartet. These are habitat loss and fragmentation, overexploitation, alien species invasions, and co-extinctions. Earth's fossil record documents 5 previous periods of mass extinction. However, because the current rate of species loss is estimated to be 100 to 1,000 times higher than natural background levels, ecologists now classify these 4 drivers, the evil quartet, as the mechanisms triggering a 6th mass extinction event. Conservation is divided into 2 distinct structural approaches. The first is in situ, or on-site conservation. This means protecting an endangered species by preserving its entire natural habitat and the surrounding ecosystem. Globally, conservationists have identified 34 regions as biodiversity hotspots, areas with very high species richness and strict endemism. 3 of these hotspots cover India's exceptionally high biodiversity regions, the Western Ghats in Sri Lanka, Indo-Burma, and the Himalaya. To manage these areas, India established specific infrastructure, exactly 14 biosphere reserves, 90 national parks, and 448 wildlife sanctuaries. You must also know the protected sacred groves, Khasi and Jaintia Hills in Meghalaya, Aravalli Hills of Rajasthan, Western Ghats of Karnataka and Maharashtra, and Sarguja, Chanda, and Bastar in Madhya Pradesh. The second approach is ex situ, or off-site conservation. This involves removing threatened animals and plants from their natural habitat and placing them in specialized settings. Key interventions include zoological parks, botanical gardens, cryopreservation of gametes, in vitro fertilization, and maintaining genetic strains and seed banks. The categorical difference is straightforward. In situ protects the overall habitat to save species contextually within their natural web. Ex situ isolates specific biological material to prevent absolute deletion from the genetic ledger. Biodiversity requires global legal frameworks. The 1992 Earth Summit in Rio de Janeiro called on all nations to take appropriate conservation measures. This was followed by the World Summit on Sustainable Development held in Johannesburg, South Africa in 2002. Here, 190 countries pledged their commitment to achieve a significant reduction in the rate of biodiversity loss by the year 2010. Finally, we must integrate the required structural morphology data from Class 11 regarding gymnosperms. Gymnosperms are plants with ovules that are naked, meaning they are not enclosed by any ovary wall. The giant redwood sequoia, shown here, is one of the tallest tree species in this group. You must accurately contrast macrostructural adaptations. For instance, psychostems are unbranched, whereas the morphology of Pinus and Cedrus features explicitly branched stems. You must also differentiate their root symbioses. Pinus roots maintain a fungal association in the form of mycorrhiza. Conversely, Cycas possesses small, specialized coralloid roots that are exclusively associated with nitrogen-fixing cyanobacteria. Whether you are parsing the legal architecture of Earth summits, calculating mathematical z-values for species distribution, or identifying the microscopic root symbioses of gymnosperms, mastering the biology curriculum requires the strict, objective categorization of all variables within the biological matrix.